Now doctor, the next favorite topic Narmada project for us is citric acid cycle, which is a multi-enzyme complex. Pyruvate dehydrogenase and alpha ketoglutarate dehydrogenase, both of them are multi-enzyme. Where do you find citric acid cycle enzymes? Mitochondria. Whereas glycolysis enzymes, cytoplasm. Yesterday we discussed one MCQ. What are all the enzymes in cytosol and mitochondria, and both of them. Urea is both cytosol and mitochondria, urea synthesis. So doctor, before the citric acid cycle begins, the pyruvate must be converted into acetyl-CoA, which is the linking part. So there is a pyruvate transport protein which carries pyruvate produced in the cytosol from the glycolysis into the mitochondria, and there the coenzyme A is attached and acetyl-CoA is being formed by the pyruvate dehydrogenase complex is what you need to basically remember. Krebs cycle occurs in which conditions? Aerobic conditions. So you should remember that how is energy captured in the citric acid cycle? In the form of NADH, FADH2, and also there is a substrate level phosphorylation where ATP is produced. But NADH, FADH2 produced in the citric acid cycle, when will they be able to create ATPs? Only if they pass through electron transport chain. An electron transport chain, who is the final electron acceptor? Oxygen. So unless oxygen is there, electron transport chain cannot function. Unless electron transport chain is there, citric acid cycle cannot function. So this is how everything is linked. So citric acid cycle may, at which step you have substrate level phosphorylation? When succinyl-CoA becomes succinate. Succinyl-CoA becomes succinate, and that is the point at which GDP becomes GTP is what you have to remember. So let us look at the various reactions, doctor. You have to be 100% sure. Agar citric acid cycle, fatty acid synthesis, beta oxidation or glycolysis, I say so-called so standard topics, doctor. Without that there is no question paper. Agar aap wrong kare to is topics mein Dr. Murli Bharadwaj soul will be heaving in graveyard. So be very sure, right? So acetyl-CoA combines with oxaloacetate to form citrate. What is that enzyme called? Citrate synthetase. You are more comfortable if I go to board or with the slide, right? So जरा white board के ऊपर हम dance करें तो much better है rather than the. Anyway, all these PowerPoint slides are available for you on the onlinembbs.com video library under every video, right? So no worries. Oxaloacetate becomes citrate by the incorporation of the acetyl-CoA. And what is this enzyme called? Citrate synthetase. Citrate typically become cis-aconitase, aconitate, by the action of aconitase. Then cis-aconitase typically become deisocitrate. Once more, it is the same aconitase is responsible. Isocitrate become alpha ketoglutarate by the action of isocitrate dehydrogenase. Alpha ketoglutarate become succinyl-CoA. Succinyl-CoA by the action of alpha ketoglutarate dehydrogenase. Succinyl-CoA become succinate, and this is called succinyl-CoA synthetase. This is the step where GTP is formed from GDP. Substrate level phosphorylation होता है. Then succinate becomes fumarate by the action of succinate dehydrogenase. Fumarate become malate by the action of fumarase. Malate become oxaloacetate by the action of mala-D. Malate dehydrogenase. So, oxaloacetate to citrate, citrate cis-aconitate, cis-aconitate to isocitrate, isocitrate to alpha ketoglutarate to succinyl-CoA to succinate to fumarate to malate to oxaloacetate. Like a parrot, you should be able to rattle out in the tomorrow's exam. Rituraj is saying board is better, sir. Slide तो कहाँ भी मिलेगा, टेक्स्ट बुक में भी मिलेगा. दिमाग के अंदर घुसना है. It should go and fix and sit in your mind.